So hi everyone, and warm welcome to this live webinar by Pelago Bioscience. We're actually here at our office here in Stockholm, Sweden, the home of CETSA that we also call it. And my name is Helena Almqvist, and I'm a senior product advisor here at Pelago. And with me I have Lawrence Arnold, and I'll let him introduce himself.
Hi there, I'm Lawrence, Head of R&D, and it's a pleasure to be here to assist with questions from our audience today. So please submit any questions via the Teams Q&A, and we'll have a session at the end of the talk.
And so with that said, it's now time to, and a great pleasure for me or for us to welcome and introduce Fiona as the speaker of today's session. She's currently heading Pharmacology and Screening at Alzheimer's Research Drug Discovery Unit at UCLA in London. And Fiona will tell us how CETSA played a key part in their project. So the title of her talk is How CETSA has been applied in lead generation delivering novel modulators of PLCγ2. So with that, I'll leave over to you, Fiona. The stage is yours.
Thank you very much, Helena and Laurent, for the very kind introduction. So I'm just going to share my slides now before we begin. So yeah, as Helena has mentioned before, I work at the Alzheimer's Research UK Drug Discovery Institute, and my name is Dr. Fiona Jeganathan, and I'm the Head of Screening and Pharmacology at the unit. So I'm going to give you a short summary as to what I'm going to talk to you about today. So I'm going to give you a brief introduction to what the Alzheimer's Research UK Drug Discovery Alliance is, and our strategy across the different drug discovery institutes, and project initiation at UCL Drug Discovery Institute, and how we sort of bring in new projects into the unit, how we assess them and bring them into our portfolio. And then I'm going to talk to you about PLCγ2, which is the therapeutic target in Alzheimer's disease, and why we've selected it as a target, and how we then applied CETSA HT to prosecute that project. So firstly, I'm going to just give you a brief introduction to the Alzheimer's Research UK Drug Discovery Alliance, just because not many people know that we exist and what we do really in our remit. So we were launched in 2015, and we were launched at a time really in the UK when pharma was exiting drug discovery in neuroscience. And at the time, Eric Karan, who was the Director of Research and Development at Alzheimer's Research UK, he was instrumental really in shaping the charity's scientific strategy and funding initiatives. And he identified an opportunity to bridge the gap between emerging academic hypotheses, lab discoveries, and clinical applications to treat patients. And he really championed the Drug Discovery Alliance being established to enable translational research. So the goal of the DDIs is really to rapidly translate new evidence-based scientific discoveries into potential treatments. And so far, Alzheimer's Research UK, which is a charity based in the UK, has invested about 60 million up to date. So in terms of what we've done so far since the inception of the DDA, is that we're 3 cutting-edge drug discovery institutes, and we're co-located in 3 academic institutions across the UK. So we're located at Cambridge, Oxford, and UCL. We're a group of about 85 scientists, 85 to 100 actually. That number is growing as we speak. Who are, tasked with looking for treatments in dementia research. We have projects collectively with over 30 pharma and biotech companies, and so far we've reached 96 million in terms of funding from external investors, in addition to our funding from our core funding from Alzheimer's Research UK. We have approximately 310 collaborations between us across 14 countries, and we're currently investigating approximately 20 targets between the 3 DDIs, for potential new treatments for dementia. So in terms of how we operate, so this is not just the UDDI, the UCL DDI, but also the Cambridge and the Oxford Drug Discovery Institute, but we operate predominantly in the preclinical development space. And our goal is, as I've mentioned, to accelerate early translation of novel targets that come out of our academic environment at 3 host institutes. We have industry standard capabilities in an academic setting, and that allows or facilitates our close partnering with the academics, and then, leveraging that to partner with pharma companies at a later stage. We've got projects that are carried out by a team of experts, and we're a mixed background between us. So a lot of us have industry experience, but also we we take from the academic environment around us, and we train them in translational research across the different core functions of medchem, screening, pharmacology and neurobiology. And we integrate emerging innovation with drug discovery with best practice to accelerate the process and rapidly take these very early scientific hypotheses into either tool molecules or something which will be preclinical and drug-like or a lead candidate. As we know, dementia is really a web of complexity, and it could be, there's multifactorial causes. So this could be involving genetic, environmental and lifestyle factors. There are diverse pathways, and these can be driven by protein misfolding, for example, amyloid or tau, inflammation, vascular issues, and many more. There's dynamic interactions, so there's mechanisms like neurodegeneration, synaptic loss and immune dysregulation, and they sort of intertwine as well. As you can see here, there's the three complex cell types that are involved, astrocytes, microglia, and the synapses. And there are challenges in treatment. So there's each form of dementia, there's Alzheimer's, there's frontotemporal and vascular dementia, and they all have unique but yet overlapping complicating interventions. So simplifying this complexity is really the key to unlocking the targeted therapies. So our strategy at the DDI, as I've mentioned, we take these early stage hypotheses from academic groups, and we do what we call target identification and validation. And we typically use the 5R principle that was set out by AstraZeneca. And this is largely done in close collaboration with our academic groups. So we will export experimental setups or assays that they've developed and sort of make them more high throughput and miniaturize and add that level of robustness, and work with them to make them fit for purpose, to adopt them for then screening, either screening a large deck and then working closely with our team of medicinal chemists at the DDI and also our team of neurobiologists, and then confirming the biological proof of concept, and then taking those to clinical development and maybe license them to companies. And I've sort of put a statistic here of how we take our hits that have got potential from our screens and turn them into leads and get them ready so that they're candidate selection. So in terms of how we take projects, so we take the root causes and we work with academics to focus on unraveling and addressing those underlying mechanisms, and we bridge the academic and industry setting. So we confirm the biology of the target, and we use our multimodal platform for screening for small molecule modulators of the target. And we then develop a suite of selectivity assays to confirm target engagement and specificity of those small molecules. We then go on to develop rodent and human cellular assays for pharmacological profiling of these compounds in their native systems. We do ADME and PK as well on these early stage leads and confirm that they're CNS penetrant, which is obviously something quite different that we do compared to other sort of biotechs that are not in this disease area. And then we run preclinical proof of concept studies in the appropriate in vivo models for efficacy and biomarkers. And then usually at this stage, we will then look to somebody to partner to expedite that process of taking it much further. So our mission is really to turn those early stage discoveries into life-changing treatments for those who are affected by dementia. So you've heard how we take our projects and kind of assess them and, really decide whether they're, something that we want to take into our portfolio. So I'm going to give you here an example of something that we did. We established that thought process and integrated this target into our portfolio. So I just wanted to add here that this is a collaborative project. So this involved actually two drug discovery institutes, so the one at Oxford and the one at UCL. And we were able to leverage the core expertise across the two units to really expedite this programme. I'm going to give you sort of the early stage part of this project, and obviously it's moved on subsequently since then, and talk to you a little bit about how we moved this project and prosecuted it across the two institutes. So phospholipase CETSA is a signalling protein, and it's involved in pathways that are critical for immune cell function and brain health. So there have been genetic insights, so that it's been well documented that there are mutations in this PLCγ2 gene, and that, such as this protective variant, which is the P522R variant. And this was identified early in a GWAS screen that was carried out in about 2015. And actually here you'll see that our principal collaborators are Matilda Catan, who's based at UCL, who's a world-leading expert on PLCγ2, and John Hardy, Professor Sir John Hardy, actually, who was part of the cohort in this GWAS study, which identified PLCγ2, this P522R variant. So we're really lucky at UCL to have access to these world-leading experts in the field. So in terms of this mutation, so this P522R mutant has been associated with reduced Alzheimer's risk, and it enhances PLCγ2 activity, suggesting that its upregulation could be beneficial. So our therapeutic strategy really is to develop small molecule positive modulators of the enzyme as a treatment for PLCγ2 for Alzheimer's disease. So in terms of its role, PLCγ2 is expressed in the microglia and brain immune cells, which are vital for clearing amyloid plaques, regulating inflammation, and also maintaining neuronal health. This regulation of microglial activity is a hallmark of Alzheimer's disease, and PLCγ2 may help restore their beneficial functions. In terms of mechanistic pathways, PLCγ2, it mediates downstream of the signaling from receptors like TREM2, which is obviously, it's well known in terms of the Alzheimer's field. And it's a key regulator of microglial activity linked to Alzheimer's. So enhancing this activity could promote beneficial microglial responses, such as improved debris clearance and controlled inflammation. And in terms of drugs targeting this, or PLCγ2, that can mimic the effects of the protective variant, boosting its activity, we could do this in a controlled manner. But we do have to be careful, because obviously modulating that pathway, as we've seen in the recent TREM2 trials, that we need to be careful that we define that therapeutic index, because if we cause excessive modulation, then we sort of go into the space of overstimulation, which could lead to excessive inflammation. So there's like a fine tuning of that signaling pathway that we need to be mindful of, and we need to design into our rationale for developing this molecule. So it's a bit of a balancing act, as I've mentioned, because overstimulation might lead to harmful inflammation, and we need to be mindful of that. So in terms of its role in Alzheimer's disease, I've got here an example from the literature. So that's the other thing we do, is we look to see where there's precedence in animal models, but also in the human evidence to support hypotheses that we're testing. So if you can see here on this slide, in the box on the left, we have a model where we've looked at the role of the P522R variant versus the sort of variant that increases Alzheimer's disease risk, and this is done in the background of a 5x FAD mouse. So in this image here, you've got two different methods for detecting amyloid beta plaques, which is the hallmark of Alzheimer's disease, and it's using specific staining or labeling techniques. So the 6E10 detects a broader range of amyloid species, including the soluble and precursor forms, whereas the X34 specifically highlights dense core plaques made of fibrillar amyloid. So essentially here you can see that in these figures where they're looking at both of these markers, when you look at the 5x FAD wild type, with the variant that increases the AD risk, you see that the levels of these plaques go up, as compared to the P522R, where you see that they're sort of at safer levels, or in this case, you see them reduced compared to the wild type. So these are sort of like the markers of the variant and what we're looking to mimic with our small molecule. And then in terms of the human evidence, there's lots of evidence in the literature, and there's the centenarian patient cohort, where these patients who live long and healthy lives, some of these patients have this P522R variant, and it's associated with slower cognitive decline when they looked at mildly cognitively impaired patients. And also in the background of the ApoE4 as well, we've seen that the positive protective effect of the variant and the detrimental effect of ApoE4 are of a similar magnitude, so they can slow that sort of accelerated cognitive decline if they have this variant, the P522R variant. So we have our sort of target validation package that then, led us to decide that this was a really good target to kind of put in our portfolio and pursue, the classical screening route. So then what we did was our team of neurobiologists, which was led by Lorenzo Magno at the time, then set about to characterize, well, what does this P522R variant do? And obviously we know that now that, increases the function of the PLCγ2 enzyme, but what does it do in the context of the signaling pathway? And so the P522R variant demonstrated enhanced activity as compared to the wild type, and this was evidenced by an increase in inositol monophosphate, IP1, and elevated levels of calcium signaling. So you can see that here, that the P522R variant as compared to the control is increased, but then you can see we also carried out experiments using the overactivated mutant, where you can see that this is also increased, but to a much higher level. But as compared to the PLCγ2, you can see that the increase is very subtle. You're looking at a fold change of maybe 1 or 1.2 or something, as compared to sort of like a 4.5-fold with the D993. So, we were very aware of the, challenges of modulating something with such a small effect. And then you can see this recapitulated with the calcium data in the figure on the right-hand side. And this was carried out in COS and HEC2 cells. So in terms of mechanism of action, so PLCγ2, in terms of the readout of the function, so we have IP1 release. So PLCγ2 hydrolyzes phosphodienol inositol 4,5-bisphosphate, so that's PIP2, to generate inositol triphosphate, which is IP3, and DAG by acylglycerol. And this IP3 is rapidly dephosphorylated to form the IP1, and it's a measurable marker of PLCγ2 activity, enzyme activity. And so we knew that actually we could configure an assay which was looking at the increase in IP1. And we know that the P522R mutation enhances this process. Increasing IP1 release is an indicator of heightened PLCγ2 function, and then also intracellular calcium release. So that generated IP3 binds to IP3 receptors on the endoplasmic reticulum, and it triggers the release of stored calcium in the cytoplasm. So we know that this variant P522R also amplifies the signaling cascade, and this results in higher cytosolic calcium. So we had two ways to measure it. So we embarked on a smaller screen, and we did this in a cell-free system, but also in the two sort of cellular markers, so IP1 and also calcium. However, we didn't have any concordance across the two screens. And then we realized that we just needed to do something a bit different in this space, because we'd run two screens historically and we hadn't found anything. And so this is where we sort of initiated this timeline at the bottom here. So we knew that we had to have on target by physical assay criteria, which was critical. We didn't really have technology that was available in-house, and we tried to evaluate by physical assays external to us, and that within the Oxford DDI as well. And we decided that actually CETSA offered an alternative approach to the types of assays that we've been configuring. It was cellular, it was thermal shift or biphysical on-target confirmation, but in the cellular context. And we initiated discussions, I think, around 2019 with Pelago, and in subsequent talks, we deemed that they had the technical components, and we were fit for purpose to prosecute this screen. And then in 2020, we initiated a fee-for-service contract with Pelago Bioscience. And we ran a fairly moderate screen with 20,000 compounds. Now, these were sort of, we curated a diversity set, and we did some modelling as well, some sort of homology modelling to identify a subset of compounds, and we compiled all these together to form this 20k compound screen and carried it out. So that sort of all took about 6 months to do. And then in terms of, So we evaluated really the CETSA HTS principles. So we know obviously that proteins have characteristic thermal stability, which is affected by many factors. And this includes binding of ligands such as small molecules. So when a protein binds to a small molecule, its thermal stability can change. So it's either stabilized or destabilized. So, and this in the context of cells or cell lysates, the cells themselves are subjected to this gradient of increasing temperatures. And as the temperature rises, we see that the protein begins to denature or unfold. So it's using this principle of protein unfolding. And these denatured proteins lose their solubility and precipitate. And these proteins remain, when these proteins are soluble, the amount of solubility is directly related to the protein stability. So we knew that if a small molecule or compound binds to the target protein, we could look to see compounds that either stabilized or destabilized, making it resistant to thermal denaturation at higher temperatures. And so that conversely, the compounds that destabilize this, the protein will make it prone to denaturation at low temperatures. And CETSA really enabled us to treat large numbers of samples. It was really a game changer because I think we were one of the first groups to really initiate this type of work, because I think the principle of CETSA had always been around, but really they reached a turning point by making it high throughput. And I think this was what made us really interested in terms of initiating this fee-for-service agreement, because we were able to run this in quite high throughput ways using the high throughput technologies like microplate readers and automated systems. And they coupled it, had a collaboration with PerkinElmer at the time, where they were using the reagents, so the alpha lyser and the alpha lyser Shore technology, which really then made it high throughput essentially. So it was proteins that were antibodies that were directed to the soluble protein of interest, in our case PLCγ2, coupled together with the streptavidin alpha donor beads, and then the capture conjugated receptor beads as well. And we knew that Pelago had already established PLCγ2 CETSA and successfully identified compounds using the label-free technology. So we were confident to set up an agreement and move forward with this screening solution. So in terms of the validation for PLCγ2, this went rapidly because, as I said before, that Pelago had already used this or used a version of this method to screen for molecules looking at PLCγ2. So in terms of validation in a cell line, we were rapidly able to select, we already had that information, so we could progress forward with the THP1 cell line. They'd already determined the melt temperature of PLCγ2. They already had highly validated antibody sets for probing PLCγ2. And they'd also already tested it as well with a small molecule that it's an annotated PLCγ2 inhibitor. However, when you run the CETSA HT in intact cells, we see a stabilization event here. As you can see on the right, the compound shifts the curve to the right. But in the lysate conditions, this curve is almost sitting on the DMSO. And so from that pilot study, they were able to determine that actually the inhibitory effect of this compound is most likely due to the decreased availability of the substrate PIP2 rather than directly inhibiting the enzyme. So we had a really nice package of data there to prosecute the screen, and we decided to move forward with our 20K set. So in terms of the screening format, we decided to go with the intact cell format because we knew that the PLCγ2 needs to be in the complex with its interacting partners, and so we thought we would have more likely be able to find hit molecules that were in the pathway, or we wouldn't limit ourselves to things that were directly on target with PLCγ2, but also in the pathway. So in terms of the format, so we pre-incubate the compounds with the cells, they're subject to a heat shock, and then we add the detection reagents. We screened the 20,000 compounds at a single concentration of 50 micromolar, and then we reconfirmed these compounds at the hits as a single concentration 50 micromolar, but in triplicates this time. And then we had 180 hits that confirmed, and then of those we were only really able to confirm or run 35 dose response or concentration response curves. So we selected those on the basis of their stabilisation and their destabilisation, so the compounds that were most stabilising or destabilising. And of those, 15 compounds confirmed. But because we had other assays in the signalling cascade or in our sort of remit, we were also able to take those 180 hits that had reconfirmed in triplicate and run them in our parallel assays. So we'd already set up this IP1 HTRF assay and then the IP1 cell-based cell-free assay as well. So that really enabled us then to kind of take compounds that were confirming across all three. So we had compounds that were on target in our cell-based cellular thermal shift assay. They were also, what we were expecting was that they would be running our cell-free, and that would also give us an indication that they were directly acting on the enzyme itself. And then we had a functional cell-based assay as well, which is obviously what we want, is a compound that then gives us the phenotype that we're looking for. So in terms of the 180, we had hits that confirmed in the CETSA and then the cell-based assay. We had hits that then confirmed in CETSA and the cell-free assay, which you can see here, the 26 hits and 62 that were in the CETSA cell-based. And then we had 39 that confirmed across all three technologies. And what we did was order all these in as solid stocks and reconfirm them. And of those 39, 23 of them confirmed as high confidence hits. So that meant if they're in the middle of the Venn diagram at the 23, they were hits in CETSA, they were hits in the cell-based assay, they were hits in the cell-free assay, and also they weren't carrying any interference liability in the assays themselves. But these assays gave us different information on the compounds. So what they were doing in the cell-based assay was that they were potentiating the signal that was already, so we used a molecule to stimulate the pathway and then add the compounds on top. And what we found was that this picked up the mechanism of potentiation rather than activation and inhibition. But in the cell-free assay, they were inhibiting in that assay that was configured. So in terms of hypothesizing, why we were seeing different mechanisms, when we were looking at this model for the phosphorylation induced, where you have these different modes of the enzyme. So when the pathway is activated, you have the enzyme sitting in this auto-inhibited form, but upon activation, the enzyme gets phosphorylated and it sort of unlatches and becomes active as it attaches to the membrane, basically what happens with PLCγ1. So the fact that we didn't identify two things. The fact that we didn't identify any direct activators in the cell-based assay indicated that the pathway needed to be activated. So we'd run that in many different modes, so we were able to detect that. And then our hits, especially the potentiators from the cell-based assay, may be missed in the biochemical assay, and that they may be activating PLCγ2 via the interaction at the membrane or its regulatory mechanism and not through the catalytic mechanism, much like the other compound we've seen historically. So after lots of rounds of ordering in compounds which were structurally similar to those original hits, we ended up with several clusters of compounds based on their chemotype. And we were sort of looking to see, how do obviously we couldn't go back and have the CETSA assay continuously through our program. But what we did was we made a decision to then go back with representative compounds of those different clusters to kind of identify whether we would observe similar shifts against PLCγ2. And so we had 10 compounds representing the different chemotypes, and then we ran it both in the intact cell format and the cell lysate, because then this would enable us to determine whether actually in the lysis method, if we're directly having an effect on the enzyme itself, or using the intact cell format, if we have the effect there and we lose it in the lysate, then it indicates we're somewhere in close proximity to the pathway. So we did that with 10 compounds, as I said before. So we reinitiated our fee-for-service agreement with Pelago, and of those 10 compounds, we saw small destabilizing effects for 4 out of 10 compounds in the intact cell format, and one of which was a stabilizer of PLCγ2 PLCγ2, but we didn't observe these shifts in the lysed cells. So the conclusion is that they're probably having their effects somewhere in the pathway or at the interaction at the membrane. So in terms of conclusions from the piece of work carried out with Pelago, so we carried out the assay in the THP1 cell line. We used an orthogonal assays in terms of our cell-based assay using anti-IgM simulation, and also a cell-free assay, which was carried out in the presence of the complex or the PLCγ2's interacting partners. We carried out a screen comprising of lots of different libraries, and it was a moderate screen. So we've got evidence that PLCγ2 can be potentiated in the cells. There's a requirement for a basal activation tone in our system. And then the differential and the directionality of the hits, we have hypotheses, and then we're using other methods to kind of perturb that, really. And that we knew that actually we made the right decision in terms of screening in the way that we did with the Pelago technology, because really through that we were able to determine actually this kind of cellular environment is key to the PLCγ2 activity, and actually it enabled us to determine that hypothesis by running the screen in the way we did, because we were able to tease it out in terms of its functional activity, but also what it was doing, on the enzyme itself. And since then, we are now at the stage where we have a lead molecule. And one of the questions that we're asking now is that, we want to do sort of a target engagement or confirmation of target with our lead molecule. So we have also been in discussion to go back to Pelago, because the other part of their business that they do is the CETSA Explore, where it's similar methods. So you incubate the cells with the compound, either in intact cells or lysate conditions, carry out the heat shock, and then run mass spec, and then conduct an analysis of the proteome. So then you have an idea of, which sort of proteins or your compound is interacting with. And they've already done this with this compound that I've mentioned before, so U73122 in the intact K562 cells. So again, that's giving us confidence that we'd be able to at least visualize the protein in mass spec, which obviously is a concern, and give us some idea about, are we directly binding, are we directly on target with our lead molecule now? But we're also evaluating other accessible in-house technologies as well in parallel. So really, I just wanted to iterate that this is kind of like a culmination of a few years worth of work, really, and it wouldn't have been made possible without the collaboration of UCL and Oxford units, and that's both the past and present team. So just to highlight Lorenza Magno, who led the project on the UCL side, and Emma Mead, who is leading the project at the Oxford side, who've now moved on to other positions, but sort of wanted to highlight those. And obviously Magda Biktash, who championed really the work with Pelago. It was an idea, and really she kind of was on board with this and really supportive. So facilitating those discussions with Pelago, and then obviously it comes with the cost, and so really championed that in terms of giving us the funding to be able to carry out this work. So I'm just going to hand over. Thank you very much for allowing me to share that story with you guys.
Thank you very much, Fiona. That was great to hear the story. And actually, since both me and Lawrence were lab active and we were doing some of the parts of this project with you back in the days in 2019, it's really nice to see how the project has evolved and the lessons you've learned, basically. But before we jump over to some questions, I just want to say that please write questions in the chat if you have any. Me and Lawrence, we have a few ones that we are eager to ask you, Fiona. So I'll let Lawrence ask the first question.
Yeah, thanks very much for a really interesting talk. I have a question on, do you consider PLCγ2 as a challenging target?
Yes. Yeah, I mean, in terms of, let's take it back to sort of, looking at it as the screening target. It's a huge protein. It's 126 kilodaltons. So, the normal techniques that you would employ, sort of biophysical, like your SPR, your ITC, double shift, they're just much more challenging to do with these multi-domain proteins. And the other thing is that where this variant is located as well, it's, I think it's it's challenging to inhibit this enzyme, that then you're looking at potentiating or activating it. So in summary, yes, it is a challenging target from that perspective, but then also where it sits in its regulatory pathway as well. You're trying to increase inflammation, really, it's a downstream effect. But, we know that obviously if we go too far, we're sort of going into the like the A-play, the autoimmune disease space. So even if we have a small molecule, it's making sure that we're careful not to hit those regions where we're tipping into that space. So it's challenging from both ends, both from a screening and prosecuting it stage, but also right through to the kind of thinking about it clinically.
Yeah, it's really interesting points. And we sort of heard it when you were describing how previous sort of more traditionally focused screens had failed to yield results. But then with quite a modest-sized library of 20,000 compounds, you've got really tractable molecules to take forward. So that was good to see. Yeah, Elena.
Yeah, so Fiona, you were an early adopter of CETSA, as you said. You were one of the first, to embark on the screening technology and to screen high-throughput screening campaigns. So what was the belief or perhaps concerns in screening a more complex environment, more physiologically relevant, in contrast to traditional screening? You mentioned that you kind of had to go to this method, but if you can elaborate a little bit more on those concerns, and again, just to, I guess, because your focus was finding modulators, and the relevance maybe was key there, but if you can elaborate a little bit more on the yeah.
Yeah, sure. I mean, when we initiated this program, we were agnostic to mode of action, really. So we were, at that time, we were looking for tools, really. So we ran our assays, with the expectation we would find something. So this is the IP1 and the cell-free assay. And obviously, when we run those screens and we didn't find anything, we were concerned, and we knew we had to do it differently, or at least find a new technology to, for hit finding, essentially. And so this really merged the two, I would say, because, we've got the target of interest, but it's in its confirmation where it has its interacting partner. So that was what appealed to us in terms of using this as a technology. So I think really it was the combination, and I think it was just that, really, in that having all of the complexities already there, and then, running that screen, really. And I think that enabled us to kind of deconvolute why we had not succeeded historically, if you see what I mean, because it was all sort of done in a roundabout way, really. We kind of needed to do the CETSA to identify that actually what we needed to do was have all the components of the complex there in the first place. And then since then, we've also done, further work to optimize the cell-free to kind of mimic that. So,
yeah, Thank you.
We have an interesting question here I'll put to you, Fiona. So do you think CETSA results could vary depending on whether cells were stimulated prior to the assay, as stimulation can, of course, influence the phosphorylation state of PLCγ2? Changes in phosphorylation status may affect protein conformation, stability and interactions, potentially impacting on the CETSA readouts.
Yeah, so I do. I mean, so is the question like why did we stimulate the cells, or?
Well, I mean, that's I guess the result of it. But do you think the results would vary depending on stimulation state?
Oh yeah, no, for sure. So that's what we've been able to do with the IP1 assay is to mimic the different modes. So you can dial in how much the tone of the system. So we've done that with triggering the anti-IgM, using anti-IgM to trigger the PCR receptor. We've been able to mimic what we think are those different states, because obviously when we're thinking about our endpoint of microglia exist in many different states. So that's what our IP1 assay is trying to mimic, that environment where it's sort of those microglia exist in a plethora of different states, really, by doing the different stimulation treatments.
Yeah, I think from a Pelago perspective as well, there's a high translation with our assays. So not only can you take to different cell lines going towards the clinic, it's also possible to run these screens in parallel. So you could have stimulated and non-stimulated states and look at those two readouts.
I mean, and that's the thing, isn't it? That the CETSA assay was obviously carried out in the TH1s. In our IP1 assay, we ran it in another a different cell line. And then obviously we've gone on to develop, secondary, tertiary assays as well, which use other cell types. And that's the definition of your molecule, taking it through all the different stages of your assay cascade to build that body of supporting evidence to know that you're actually, exhibiting the phenotype in a far removed system versus what is it doing in your physiologically relevant system. So, and that, that's data I haven't shown today. So.
Absolutely. And I have a further question here about, can you speak a little more to the paradoxical effect of inhibition in the biochemical assay versus activation in the cell-based assay? And further on, can you reproduce that in the cell-free assay by testing controlling the phosphorylation state of PLCγ2?
Yeah, we saw inhibition in that assay was because, like I said before, that we think that the enzyme exists in a state which isn't, in that it's sitting in that sort of autophosphorylated state, which doesn't allow for this molecule to bind. So we have done substitute. So actually, this has mainly been driven by the Oxford team. So the split really has been cell-based assays at UCL and cell-free assays at Oxford. So they've really worked hard to develop a really sophisticated cell-free system to mimic what we do in the cell-based system, which I haven't gone into details about because obviously, this is still an active ongoing project. So.
And perhaps a question for you, Elena, about did Pelago employ a counter screen for assay artifacts of the assay readout, or alpha readout, sorry?
Absolutely. So that was part of this project, and that was also part of all our projects we run in the hit triage. After HDS, when we do a full dose response, we always do a counter screen to eliminate assay artifacts or non-CETSA effects, as we say, that is, compounds could interfere and do something else with the protein that is a non-CETSA effect. So that is always, we always run counter screens along with the data. Yep.
Yeah, just to reiterate, I did say, like in the 23 hits, interference was one of the criteria that we included to keep compounds in that.
Yep.
And Fiona, I wonder could you comment anything about if there was a common feature in the compounds? If you're coming from a cell-free assay, of course, cell penetrance is an issue here, or could be an issue when you're going further into cell-based assays. Was there any of those considerations in play here?
So, at that stage, we couldn't really make any conclusions about the structure because obviously it was a diversity set. So the idea is that, they're as structurally diverse as possible. And then we did, we were after the rounds of SAR. I'm avoiding saying nearest neighbours because our head of chemistry hates that term. So we ordered in about, close to sort of 500 compounds that were, structurally similar to do SAR on, and then we ended up with clusters of compounds. So then they would cluster into different chemotypes, which then, some chemotypes were reconfirming in the cell-free, some were confirming in the cell-based, and then we had, compounds that were hitting the different assays.
That's nice to hear. And I was going to say if you employ, but really want to say when you employ CETSA in your next project, after knowing what you now know and the experience, would you change any aspects?
In terms of the interaction, it was one of the most straightforward. It was literally, I think we worked on getting the quotes together, and that, that was, I think that contract back and forth, numerous times. So in terms of the interaction, that was really straightforward. In terms of the workflow as well, I think we had a plan in place and then COVID hit. So we then had to change our strategy slightly and we adapted. I think we had to ship compounds to you guys, and you were able to prosecute that assay at the time where our labs were shut down. So that enabled us to continue our project forward, really. Otherwise it would have, down to a stop. So that for us was valuable in that we could still prosecute our program and continue with our workflow.
Thank you,
so
Fiona.
Would I change anything? No.
Thank you,
and in
Fiona.
terms of the data analysis, it was really, we would have what would you call it, a data release meeting, and that would be like a formalized deck with all the data. And then I think we went back and forth as well with the data analysis. So, I think I needed the raw data. We needed to look at it, do various versions of analysis. So it was straightforward as well.
Thank you, Fiona. And I think we don't have any more questions in the chat, so we'll just have one final question. And I think you have already answered some of it in your last sentence here. But if you, in general, can describe the collaboration, the experience of collaborating with Pelago, what would that be? How would you kind of summarize and conclude the project so far? We would love to do more projects with you, of course, but if you summarize your experience, how it was working with us.
I mean, this is, I think, the difference between working with a company where the technology has been born out of the company, because, they are the experts in the technology. So it was one of the most straightforward collaborations, because it was simply a case of, we prescribe what we want and you guys go away and do it. And then we get a really nice package at the end. And that's, I think it's the difference between something where, you are outsourcing a piece of work or asking somebody to do something where the expertise isn't there. So yeah, it was just straightforward, easy to do, and really enjoyable working with you all.
Yeah, thanks, Fiona. Same from us, from whole Pelago. And with that, I guess it's a wrap. And again, thank you very much, Fiona, for taking the time and present this story for us. And also, thank you very much for dialing in or listening in. And stay tuned for our next webinar. We will for sure come back. So thank you very much.